Hello and welcome to the HIVRNAtestguide.com YouTube channel, your online partner for quick, affordable, and 100% confidential HIV testing. Whether you're looking for quick, confidential results or simply want to stay informed about the latest in HIV research, you're in the right place. For more information, visit HIVRNAtestguide.com. Check the link in the description below or visit the bio section of our channel. You can access our more than 4,500 HIV testing labs across the United States, just a click away. So, without further delay, let's start today's video.
Welcome back to the Deep Dive. Today, we have a genuinely remarkable stack of sources in front of us.
We really do.
And we're not just talking about, small incremental steps forward in science.
No, not at all. These are pointing toward potential paradigm shifts.
Oh.
Really fundamental changes in how we think about things.
Exactly. Across two completely different, but equally critical areas of health. So we're going to do a real dual topic deep dive today.
One on prevention and one on treatment.
High precision prevention and what you could call treatment optimization.
It's a really exciting moment, actually. The data we've been looking at shows they're just skillfully leveraging decades of basic science.
All that foundational work.
Right. Especially our understanding of immunology, which has just exploded.
Yeah.
And they're combining it with these transformative new platforms.
Like mRNA, which everyone is familiar with now.
Exactly. mRNA in particular. And they're using it to solve problems that have honestly seems almost intractable for decades.
So what's the common thread here?
I'd say the overarching theme is precision. Just incredible precision, using molecular level targeting to get the absolute maximum medical effect.
While minimizing the burden on the patient.
Minimizing the physical burden, the time, the logistics, all of it.
Okay, that concept of precision really sets the stage perfectly. Our deep dive is built around two main pillars. The first one is, it's arguably the biggest news.
I would agree.
It's the astonishing progress in HIV vaccine research. We're talking about early, but very promising, results from the first human trials of a completely novel strategy. It's called germline-targeting.
And then, for the second pillar, we completely shift gears over to cancer care.
Right.
We have some really compelling clinical evidence, and this is published in top-tier journals, showing that for certain patients, and the selection part is key.
Okay.
For specific patients with prostate and cervical cancers, they can get the exact same excellent long-term outcomes with treatments that are just significantly shorter or less invasive.
So the whole less is more philosophy.
But validated by hard data, which is what matters.
Yeah. All right, let's unpack this and set our mission for this deep dive. We have to start with the core science behind this germline targeting for HIV.
It's complex.
It is. So we need to simplify the biology of B-cell activation so you can really grasp the genius of it, but also the sheer difficulty.
And then we'll pivot to those cancer trials.
Yes, and understand how that data from places like JAMA Oncology could fundamentally simplify and improve cancer care for thousands of people, maybe starting right now.
So when we're done, you'll walk away with the key insights, the implications. Really, the whole picture of medicine moving from sort of blunt instruments to molecular scalpels.
Let's jump in.
To really get this first part, you have to start with the fundamental problem. The reason HIV has been such a frustrating target for immunologists.
We've been chasing a vaccine for, 4 decades now?
About that, yeah. And the core challenge is exactly the same as it was at the beginning. HIV is a master of disguise.
It mutates constantly.
Constantly. Specifically, it's that outer coating, the viral envelope protein. It changes its shape so dramatically, so quickly, you can get hundreds of distinct strains inside a single infected person, let alone globally.
So that envelope is the real problem.
It's the crux of the issue, yeah. See, with a normal virus, your immune system develops antibodies that are perfectly tuned to that virus's exact shape. But with HIV, the virus infects a cell, and the first thing it does is basically shed that coat and put on a new one for the next round.
So a traditional vaccine is training your immune system to hit a stationary target.
And HIV is throwing 100 moving targets at you at once. The immune response you get is often really effective, but it's narrow.
Meaning it only works on the one version it was trained on.
Exactly. And it becomes useless the second the virus changes its coat. The immune system is just constantly playing catch-up.
So the goal can't be to make antibodies against just one strain.
No, that will never work. You need antibodies that can neutralize a broad range of strains. And these have a name. They're called broadly neutralizing antibodies.
Or BNAbs, for short.
Right, BNAbs. And for decades, scientists have known they exist. They've found them in a small percentage of people who have lived with HIV for a long time.
But we couldn't figure out how to make a vaccine that could reliably get a healthy person to produce them.
Exactly. We knew the destination, the BNAB, but the map we were using was wrong. And that brings us directly to the breakthrough strategy in these new reports, germline targeting.
Okay, let's break this down because it sounds complicated. What does it mean to target a B cell in its germline state?
It is complicated, so an analogy is probably best here. Think of your B cells. Those are the cells that make antibodies. Think of them as a massive library, a library full of weapon blueprints.
Okay.
Most of these B cells are in what we call a naive state, the germline state, meaning they just hold a very rough, basic blueprint. They have the potential to become a weapon, but they're not specialized yet. They're just dormant.
And most of them aren't the right ones for HIV.
The vast majority. When they see HIV, they're destined to produce narrow, ineffective weapons. They're like a blueprint for a slingshot, when what you really need is a guided missile.
So the normal immune response is just grabbing thousands of those slingshot blueprints and hoping one of them gets lucky.
That's a perfect way to describe it. Germline targeting is a completely different strategy. The goal is to bypass all those millions of ineffective blueprints and find the one very rare naive B cell.
The one in a million.
Literally, the one that has the absolute best starting genetic code to eventually evolve into a cell that produces that multi-tool weapon, the BNAB.
So you're not just triggering a general alarm and seeing who shows up. You're proactively going in, finding a specific dormant cell, and waking it up.
You're guiding its evolution. That's the core concept. You use a highly engineered piece of the virus and immunogen to activate these B cells while they are still in that naive germline state.
And why is it so important to get them that early?
Because if you don't, they get outcompeted. The millions of other B cells that make the useless slingshot antibodies wake up faster and louder, and they drown out the signal from the one you actually need. So you have to prime them first.
That sounds incredibly difficult. It's like finding a single specific grain of sand on a beach and telling it, okay, you're the one, start growing into a supercomputer.
It is. And the vaccine does this with a very deliberate, staged approach. That's why the sources call it a prime and boost sequence.
It's not a one-and-done shot.
Definitely not. It's a guided evolution. Step 1 is the priming. The vaccine delivers a specially designed HIV immunogen-tase, a small synthetic fragment of the virus protein.
And this fragment is designed to do what exactly?
Its only job is to be the perfect molecular key that fits the lock on only those rare precursor B cells.
So it's not trying to stop the virus itself.
Not at this stage. It's just an alarm clock. It finds those 1 in a million cells, wakes them up, and basically tells them, you're up, start evolving for this mission.
And once that cell is awake, or primed, it's still not ready to make the final weapon, right?
Not even close. It's awake, but it's not a mature warrior yet. That requires the second stage, the subsequent doses, the boosting.
This is the guided maturation part.
Exactly. Once those B cells are primed, they start a process of rapid refinement. And you give them subsequent booster shots with slightly different or complex immunogens. Think of it like a series of increasingly difficult exams.
You're training them.
You are pushing them with each dose to refine their antibody structure, bit by bit, until they finally mature into cells that produce the real deal. The effective BNABs, they can hit that whole spectrum of viral variants.
And this is where it gets really exciting, because the sources are reporting that this actually works in people.
The first step does, and that's a monumental achievement. The early results from 2 separate research groups published together in Science confirm that the priming phase finding and waking up the right cells is successful.
This isn't just in a petri dish.
No, this is a validated human immune response in a clinical trial.
One of the senior investigators, Rogier Sanders, had a quote that really stood out to me. He said they saw an immune response that indicates that we're on the right track.
He did, but it was the next part of his statement that made everyone in the field just stop and listen.
The part about precision.
Yes. He said, we saw that we can target the cells that we need to target with atomic precision.
Atomic precision. I mean, that's not a term you usually hear in biology. It sounds more like engineering.
And that's the point.
Yeah.
It means the immunogen they designed and delivered was engineered with such exquisite, detailed specificity that it only locks onto the receptor of the exact precursor B cells they wanted.
So it ignores all the other billions of B cells.
It ignores them completely. Imagine the receptor on the B cell has a very specific keyhole shape determined by its genetics. The immunogen is built, atom by atom, to be the only key that fits that specific keyhole.
And that precision is so important because if you triggered a general response,
You'd flood the system with all those slingshot antibodies. They would just create noise and disrupt the guided evolution of the rare cells you actually want to train.
So getting that clean start is everything.
It is. It validates the entire theory. But of course, Sanders was also realistic. He was quick to point out the next major hurdle.
Right. He said now that they've successfully primed the cells, they found their trainee, the next critical step is to further stimulate these cells to secrete broadly neutralizing antibodies.
And that's the whole ballgame. Priming is step one. Getting functional BNAb secretion is the ultimate goal. But we can't overstate how important this first step is. It proves the blueprint is valid. Now it's about perfecting the training regimen.
And speaking of technology, it's fascinating how this deep immunology connects directly to the vaccine tech that we've all become so familiar with recently.
It's really significant that these reports covered two separate approaches that both worked.
Right, it wasn't just one lab. It adds a lot of weight to the finding.
It does. And this second team of researchers, they use a platform that everyone will recognize.
They use mRNA.
Yes, specifically they used mRNA-encoded nanoparticles produced by Moderna to achieve that same successful priming of the germline B cells.
Which immediately shows how much we've benefited from all the accelerated research during the pandemic.
Absolutely. Moderna's system is basically a delivery service for instructions. The mRNA tells your body's own cells how to produce the specific protein fragment, the immunogen that's needed for the B cell training.
So instead of a drug company spending years in a lab trying to manufacture these really complex designer proteins,
And purifying them and stabilizing them.
you just inject the blueprint, the mRNA code, and your own body becomes the factory.
That's the key. And it provides a massive advantage, especially for a multi-stage process like this one. The single biggest benefit here is speed.
The sources were clear on that. Using mRNA would allow for significantly faster vaccine development.
Think about it. For the boosting phase, you'll probably need to test lots of slightly different immunogens to get the maturation just right. With mRNA, if you need to tweak the booster, you just rewrite the code on a computer. You can have a new formula ready for manufacturing almost instantly. You completely bypass that slow, expensive process of making a new physical protein every time.
That flexibility is a game changer for clinical trials.
It's unparalleled, especially for a global health problem like HIV. You need speed, flexibility, and scalability. mRNA delivers on all three.
Of course, you have to report on the full picture. The sources did note one small drawback in these early trials.
They did. It's important to be balanced. While the priming worked incredibly well, a small number of the patients who got the mRNA versions had skin reactions at the injection site.
Okay, were they serious?
No, they were described as mild and self-limiting. But any side effect, no matter how small, has to be monitored very closely, especially for a vaccine that you'd hope to give to millions of people.
And they'll need to figure out what's causing it.
Right. Is it the lipid nanoparticle that carries the mRNA, or is it the HIV immunogen itself? They'll need to optimize that.
But even with that minor issue, the flexibility of the mRNA platform showed up in another really important way.
Yes, in the geography of the trials.
One trial was in the US, but the other one, critically, was conducted in Rwanda and South Africa.
And that choice of location is not just a detail. It is scientifically and contextually vital.
Because that's where the need is greatest.
Exactly. The majority of people living with HIV are in Africa. So you absolutely have to prove that your vaccine concept works in that population.
And this kind of cutting-edge approach, germline targeting, it had never been tried there before.
Never. The sources make that clear. This was a first. And it's so important because people's immune systems can respond differently to vaccines based on a whole range of factors.
Like genetics or...
Genetics, diet, other infections that might be common in the area. You can't just assume a vaccine tested only in North America will work the same way everywhere else. You have to do the work.
So what did they find? Did it work as well in the African cohorts?
The finding was overwhelmingly positive. The researchers reported that the mRNA approach appeared to work just as effectively. It achieved that necessary B-cell priming in both the North American and African populations.
That's huge. It suggests the biological pathway they're targeting is highly conserved across human diversity.
It does. It means that the molecular key fits the lock, regardless of the patient's background, at least in these initial trials.
And what does that mean for the next steps?
It means the door is wide open to move forward. To start testing the full prime and boost sequence, specifically for African populations in most need of an HIV vaccine, it's a massive step towards equity in vaccine development.
What a powerful note to end that section on. So now let's make that pivot we talked about, because that same philosophy of maximizing efficacy while minimizing the burden on the patient,
Showing up in cancer treatment, too. It's incredible.
from prevention to treatment optimization. Let's shift gears to oncology.
So we're moving from this very long-term fight against a virus to making immediate improvements in how we manage existing cancers.
And the sources here point to two studies that really offer hope for de-escalating therapy, which is such a critical conversation.
It is. I mean, for so many people, cancer treatment means months of grueling schedules, financial strain, severe side effects.
So the ability to de-escalate, to do less, but without sacrificing the outcome.
That is the holy grail of modern oncology, without a doubt. Every reduction in treatment intensity, fewer radiation sessions, a less extensive surgery, it all translates directly into a better quality of life for the patient.
So let's start with prostate cancer.
Yes. The first study was in JAMA Oncology, and it focuses on a very specific group of men. These are men who have already had a radical prostatectomy, the biggest surgery for prostate cancer, and now they need follow-up radiation.
Just to catch any microscopic cancer cells that might have been left behind.
Exactly. But the conventional treatment for that is it's a slog. It's low doses of radiation delivered daily.
Every single weekday.
Every weekday for up to 7 weeks. That's around 35 separate trips to the hospital.
7 weeks. I mean, just imagine trying to hold down a job or live your life while doing that. It's a huge burden.
It's an enormous barrier to care for so many people. Now, the new approach they studied is called stereotactic body radiotherapy, or SBRT.
And the big difference here is the schedule.
A massive difference. Instead of all those low-dose treatments, SBRT uses high-precision beams to deliver a much higher, more potent dose of radiation. And it's all done in just 5 sessions.
From 7 weeks down to 1 week.
The clinical appeal is just overwhelming. But there was a reason, a historical hesitation, for why it wasn't widely used in this specific situation after surgery.
What was the concern?
It really came down to anatomy. After you remove the prostate, the area you need to target, the prostate bed, it can shift around a bit more.
Because of normal body functions,
like your bladder filling up or gas in your bowel, things that happen every day. And with SBRT, you're using these really high doses, so you need absolute sub-millimeter precision.
because you do not want that high dose hitting healthy tissue.
Especially not the rectum or the bladder, which are right there. The fear was that you could cause really severe life-altering side effects. Chronic bleeding, incontinence, things that destroy a person's quality of life.
So the study was designed to see if modern SBRT, with better imaging and guidance, could overcome that risk.
That was the exact question. They took 100 men, treated them with this 5-session SBRT, and then tracked them very, very closely.
And what was the outcome?
The outcome was fantastic. 2 years after the treatment, their cancer control rates were just as good as what you see with the long 7-week course.
And crucially, what about the side effects?
Also similar, they didn't see an increase in those severe side effects that they were worried about. So no sacrifice in safety or efficacy, but a monumental reduction in the treatment burden.
One of the study leaders, Dr. Amar Kishan at UCLA, really summed it up.
He did. He said that if these results hold up in longer studies, this approach could remove a major barrier to post-surgery radiation therapy.
And that's not just a convenience. For some people, that's the difference between getting the treatment they need and not getting it.
It makes the best care more accessible. It reduces the financial toxicity of cancer, careless time off work, less money on gas and parking. It's a huge, immediate win for patients.
Okay, so from reducing time to reducing invasiveness. Let's pivot to that second oncology study on cervical cancer.
Right, so this second study, published in JAMA Network Open, is all about surgical de-escalation for women with cervical carcinoma.
And again, the sources stress that this is for a very specific group. We have to be really clear about that.
Absolutely. This isn't for everyone. This finding only applies to carefully selected patients with low-risk early-stage cancer, specifically stage IA2 or IB1.
And they're being treated at major cancer hospitals.
That context is everything. These patients have the best prognosis. The cancer is small and localized, so the risk of it having spread is minimal, but you can't be sure it's zero.
And historically, the approach was to be very aggressive surgically.
Right, the standard was often a radical surgery, just to be absolutely sure you got every possible stray cell. So what these researchers did was compare the outcomes for 3 different levels of surgery in this specific low-risk group.
Okay, what were the 3 options?
The least extensive was a simple hysterectomy. That's where you just remove the uterus and cervix.
But you leave the surrounding tissues.
Right. You leave the supportive ligaments, the nerve bundles, all of that intact. Then they compared that to a modified radical hysterectomy, and finally to the most extensive, the full radical hysterectomy.
And that radical surgery takes out a lot more tissue.
A lot more. It removes a wide margin of the tissue surrounding the uterus, the perimetrium, and more lymph nodes. And while that gives you the maximum theoretical protection, it also causes the most collateral damage.
Which means a longer, more painful recovery.
And a much higher risk of permanent complications.
Mm-hmm.
Things like bladder control issues or lymphedema, because you're damaging nerves and lymphatics that you just can't avoid.
So the big question was, for these low-risk patients, is that radical surgery overkill? Are we overtreating them?
Precisely. And they looked at a huge number of patients, over 2,600. The finding was incredibly powerful.
What was it?
They found no statistically significant difference in survival rates between the three groups. None.
So doing the biggest surgery didn't make patients live any longer?
Not at all. And that held true over the long term. At 3 years, 5 years, 7 years, and even 10 years after the surgery, the survival was the same.
10 years is the benchmark, isn't it? That's when you really feel confident about a cure.
It is. And on top of that, there was no difference in post-operative outcomes either. So doing less surgery didn't create new problems.
Well, the implications for patients are just enormous.
Huge. We can now confidently say that for this specific low-risk group, a simple hysterectomy is enough. The benefits are so tangible.
A shorter recovery time, less risk from the surgery itself.
Lower costs, less time in the hospital, and most importantly, a much lower risk of those devastating long-term complications.
Like the bladder issues you mentioned.
Exactly. That's often the biggest one. With a simple hysterectomy, you preserve those critical pelvic nerves. The patient gets the cure, but she also gets to keep her normal quality of life for the decades to come.
It's the same theme we saw with the prostate cancer study, using data to refine our standards to stop doing things just because that's how they've always been done.
It's optimizing care not just for survival, but for wellness. And it all connects back to that idea of precision, precisely selecting the right patient for the right and sometimes smaller intervention.
So this has been a really expansive deep dive, all the way from the absolute cutting edge of immunology and prevention
To the immediate practical optimization of cancer treatments we use every day.
It's pretty rare to see such clear wins reported across such different fields all at once.
It really is. And when you pull back and look at all of it, the theme that just jumps out is this relentless push for efficacy plus efficiency.
And accessibility.
And accessibility, yes. Whether it's the HIV vaccine using mRNA to slash development timelines, or the cancer studies cutting a 7-week radiation course down to 5 days.
It's all about improving the patient's experience and safety without compromising the outcome.
It's data science meeting biological precision. And the goal is to lessen the burden of managing disease.
Okay, so let's nail down the key takeaways one last time for you. First, on the HIV front, we have seen this theoretical idea, germline targeting, actually work in humans.
Using atomic precision to wake up the exact right B cells needed for the job, it proves the blueprint is viable.
Second, mRNA technology is showing its value far beyond the pandemic. It's speeding up complex vaccine development.
And crucially, it's making it possible to run these trials in the places that need the solutions most, like Rwanda and South Africa. It's building in global equity from the start.
And finally, in oncology, data is helping us treat cancer smarter, not just harder.
High-dose, short-course SBRT is proving to be just as effective and much more patient-friendly for certain prostate cancer patients.
And for carefully selected women with early-stage cervical cancer, a simple hysterectomy offers the same excellent long-term survival as a much more radical surgery.
It's a move toward a high-precision, low-burden model of care. And the implications go way beyond just these two diseases.
That's what I was thinking.
These reports really confirm that this combination of deep biological understanding and rapid prototyping technology like mRNA is creating totally new ways to solve problems that seemed impossible just a few years ago.
So we want to leave you with a final thought on that, a question to mull over. We've seen mRNA work as this universal instruction platform, right?
To make designer immunogens for HIV,
Yeah.
to make a rapid response vaccine for COVID.
So if this technology can so precisely instruct our bodies to build these incredibly complex training molecules for the immune system, what else could it do?
What other long-stalled medical challenges might this platform suddenly revolutionize in the next, say, 5 years?
Could it be used for therapeutic protein replacement Or what about training the immune system out of harmful patterns, like in autoimmune disorders?
Keep an eye on this technology. Its applications are just going to keep expanding. We are truly at the beginning of understanding its full power. The age of molecular precision is here.
Thank you for joining us for the deep dive. We'll see you next time.